Hello, everyone, and welcome. I am happy to host today's webinar, Polymer Screening for Amorphous Solid Dispersions using the Crystal16 instrument. This is a webinar presented by Thierry Bonnaud from Veranova. But before we go into more details and I allow Thierry to present this case study, I would like to let you know that the webinar is being recorded and it will be made available in the coming days. You'll receive an email with information where you would be able to re-watch this webinar or download the slides of the webinar. If you have questions, please send us your questions to info@crystallizationsystems.com or make use of the chat function, question function of this platform and write to us your questions there. We will take questions at the end and we will be able to answer them together with Thierry. Will last about 45 minutes, this webinar. And for those that do not know Technobis Crystallization Systems, I would like to introduce you our company. We are a Dutch company located in the north of the Netherlands, world-leading technology provider for solid-state research, process development, and formulation. In our portfolio, as you can see, we have actually two portfolios, the crystallization systems, as you see here, with the Crystal16, Crystalline, and Crystal Breeder instruments. I see some names attending today. Some of you already know our instruments, and a new product line, the reaction screening with our Reactol introduced early last year. About the Reactol, it is an multiple reactor system with 5 reactors in parallel that you can heat and cool in a controlled way from -10 to 150 degrees. And the beauty of it is the automated sampling. Your samples are, while you are performing your reaction, you can sample, quench, and immediately dilute, directly injected in an HPLC vial. So after a few days or after a few hours, you can go back to your instrument, take your tray with the HPLC vials with your samples that are quenched and already diluted and analyzed by LC-MS. Is the instrument for reaction screening, optimization, kinetics, and so on. And more related to today's webinar, our crystallization portfolio with, as I mentioned a bit earlier, you might already know the Crystal16 and the Crystalline. You will hear more from Thierry about the Crystal16 instrument. Remember, the Crystal16 is your instrument for solubility, metastable zone with solid-state screening, and the Crystalline with the camera technology that works exactly like a microscope. Also Raman. If you have a Raman spectrometer in your lab, you can easily connect it to the Crystalline. Will help you to develop and optimize your crystallization and formulation processes. Crystal Breeder with its vapor diffusion setup and controlled evaporation, another gadget, is this instrument that will help you to grow single crystals, perform crystallization screens at very small scale. So for very early discovery, when not that much material is available, this is the ideal instrument to use. And with that, I want to go to our and introduce you our speaker today, principal scientist Thierry is principal scientist solid form and particle engineering at Veranova. He has a background in organic synthesis and drug discovery and over 27 years of experience in the industry. He worked at the Neuroscience Center of Pfizer in 1999 in Cambridge, UK, and then joined in 2000 the combinatorial chemistry group at Medveer, Cambridge, where he was involved in synthesis of new enzyme inhibitors. He joined Amorphous, part of SAFC Pharma, Cambridge in 2007, and has continued to work at the site, which today serves as a global center of excellence for solid form and particle engineer at Veranova, a global CDMO. During his time, he has had several roles of increasing seniority, spanning project and technical leadership. His technical interests span across salt polymer screening, co-crystal studies, including chiral separation, and he is the author of review on co-crystals and co-author of more than 20 patent applications. And with this, I will actually stop my sharing, and I will give the floor to Thierry.
Hi, my name is Thierry Bonnaud, and today I'm going to talk about polymer screening for amorphous solid dispersions, and this is using the Crystal16. So let me take you through the history of Veranova, the company I'm working for. So Veranova started as a name in 2022 after the acquisition of Johnson Matthey Health Business by Altaris. But it goes back 200 years ago with the creation of McFarlane in Edinburgh. I have put here some of the milestones and different acquisitions, including the acquisition of the Amorphous business created in 2003, and that acquisition took place in 2017. And I'm part of the Amorphous business. Veranova counts over 700 employees. We have different sites, 2 in the UK, a manufacturing site in Edinburgh, and an R&D site in Cambridge, specialized in solid form. We are a CDMO with other sites in the US, such as the site in Devon, which has been FDA inspected recently, and we have other sites, North Andover and West Deptford as well in New Jersey. I am based in the Cambridge site, which is based in the UK. We have offerings covering the development of drugs, all the way from candidate selection through solid form development and particle engineering. Nanosuspension is part of enabling formulation, like micronization, amorphous solid dispersion, and these are technologies to boost the performance and improve stability for challenging drugs. But we have many other services, as you can see here. Our solid state services operate under the brand Amorphous. We have worked over 2,500 projects since 2003 when we started. We have worked on over 15% of small molecules which have been approved in the last 10 years. We get also to work on more and more complex molecules, including proteins and peptides. In this presentation, I will begin by introducing Piroxicam, our model compound for the study. I will then explain what amorphous dispersions are and discuss the excipients that can be explored to prepare them. After that, I will walk you through the principle of the solvent shift method, a valuable tool for polymer screening, and show you how we adapted this method for the Crystal16 to work, especially with Piroxicam. Finally, I will present some of our amorphous dispersion preparation, along with the characterization and the final results. Before diving into our study, let me introduce you briefly to Piroxicam. So Piroxicam is a well-established anti-inflammatory drug. It was first introduced to the market in the early 1980s and has been widely used ever since for the treatment of conditions like pain and inflammation. It can be taken orally, in capsules or tablet form, which typically has a daily dose of 20 mg per day. Despite its long history of use, Piroxicam still attracts interest in research because of its poor water solubility, which can limit its absorption and effectiveness. This makes it an excellent model compound when studying new formulation approaches. From a polymorphic point of view, the landscape of Piroxicam has been well studied. In fact, there is a key paper from 2012 which established 3 distinctive anhydrous forms as well as a monohydrate. In-house characterization, we looked at the amorphous phase, which has been found to have a low TG and tend to recrystallize very rapidly. Now, another important feature of this molecule is that it contains multiple ionizable centers, the most relevant being a weakly basic pyridine and an acidic phenol group. And when we compared with our experimental measured pK against the calculated value from Percepta, we can see very small but noticeable differences of around 1 unit between the experimental that we collected in-house and the predicted values. Because Piroxicam has multiple ionizable centers, its behavior in solution depends strongly on the pH. In this figure, you can see the distribution of species. Up to 3 different forms of a molecule can exist in equilibrium depending on the pH. At very low pH, below 1.9, the protonated species dominates. Between roughly 1.9 and 5.3, the molecule is mainly in its neutral form. And once the pH rises above 5.5, the deprotonated species become the most abundant species. What's important here is that each of these species has a different solubility. That's why we perform additional experiment measurement across a range of pH values to capture this behavior more accurately. As part of the solubility profiling of the molecule, we started by exploring different media compatible with IV delivery. We measured the solubility by HPLC after 24 hours equilibration, and we can confirm that the solubility of Piroxicam is likely to be too low for an IV administration unless some solubilization enhancing formulation is used. On this slide, we continue our investigation, this time for the solubility in various media and buffers commonly used for oral delivery. As expected, solubility was quite limited at low pH, but increased progressively at pH approaching 6.5 and neutral. Notably, solubility was enhanced in passive media, and we observe a visible color change, which we trace back to solid form transformation, from the yellow form 1 to the white monohydrate form of Piroxicam. And this transformation is reported in the literature below. Now, let's take a look at Piroxicam through the BCS solubility classification, which is widely used across the industry. This framework classifies drugs based on their solubility and permeability, and this can help to predict their in vivo performance. Class 1 and 3 compounds generally dissolve, while class 2 and 4 pose more a challenge in terms of viability due to their low solubility. At a dose of 20 milligrams, Piroxicam is practically insoluble at pH 4.5, which placed it firmly in BCS Class II. As you can see, Piroxicam has a real potential to benefit from solubility enhancing technologies. When starting a project, we always begin with batch characterization to confirm material purity and established a baseline for comparison. This characterization includes solid-state methods, such as XRPD, to assess crystallinity and polymorphic form, thermal analysis, and purity checks by HPLC and NMR. So for Piroxicam, we are working with a crystalline form 1, which has a high purity and a melting point of approximately 201. The amorphous phase, by contrast, has a potential for higher solubility, and in our case, it was generated using our DSC instrument through a melt quench cooling. However, due to the low glass transition of around 48 degrees, this amorphous phase is likely unstable and prone to crystallization, and will require a polymer stabilizer to maintain its amorphous state. Now, let's dive into the exciting evolution of solubilization technology over the last 25 years. Over 5 years ago, Pharmaceutical conducted a survey to evaluate these approaches in marketed drugs. The data clearly shows how dramatically the landscape has changed. While amorphous dispersions were relatively uncommon 25 years ago, they have now surged ahead of all the other technologies, with more than 40 products on the market. Lipid formulation and cyclodextrin complexes remain widely used, but since 2014, amorphous dispersion have seen a remarkable boost. This rise likely reflects the maturation of this technology that was new in the early 2000s and now has proven its full potential. So now, how do we go about selecting the right technology? The answer really depends on the specific property of the API. At Amorphous, we have developed a formulation workflow. The physical property of the API can guide us on the choice of the final formulation. This workflow includes a wide range of technology such as salt formation, co-solvent screening, inclusion complex and co-crystals, nanosuspension, lipid formulation, and amorphous dispersion. For Piroxicam, the key parameters such as the log P, the dose, and the melting point really strongly indicate that ASD is a promising technology to pursue. So what are amorphous solid dispersion, or ASD? In an amorphous solid dispersion, the drug is stabilized in an amorphous state within a polymer matrix. This dispersion helps to prevent crystallization, improve dissolution, and lead to a better drug absorption compared to the crystalline form. The key benefits are faster dissolution rate, higher apparent solubility, and a longer period of supersaturation. Unlike the pure amorphous form, which often shows a spring parachute effect, the right polymer can help sustain that supersaturation. It also plays a crucial role in keeping the material amorphous during its shelf life. So, what can we actually use to build this stabilizing matrix? In practice, we rely on approved pharmaceutical excipients, most often polymers. These are available in different grades with different glass transition temperature. Some are purely synthetic, such as PVP, PVPVA, and polymethacrylates. PVPVA is a cross polymer and is one of the most popular for commercial ASD. SoliPlus have been specially designed to improve drug solubility, but as far as we know, there is no commercial product using this polymer yet. We also have semi-synthetic option, particular cellulose derivatives such as HPMC-AS, which is widely used commercially. Factors such as drug polymer miscibility, the polymer's TG, the potential for hydrogen bonding, or ionic interaction also play a role in determining which excipient is most suitable. Proteins and other large molecules can also serve as an alternative to polymers. While there are studies in the literature exploring this approach, we have not seen yet any commercial products using this excipient, but this is definitely a place to explore. Interestingly, many of these excipients have been used in the food industry for quite a long time, and now some manufacturers are exploring them under GMP and pharmaceutical grade standard to make them suitable for ASD application. Another exciting development in recent years is the use of small molecules, such as amino acids. These systems are known as co-amorphous dispersion, and they are typically designed in a simple one-to-one molecular ratio with a drug. The idea here is that the small molecules interact as stabilizing, forming a strong molecular interactions with the API. And while this is still a relatively new area, co-amorphous dispersions are gaining attention as they often allow higher drug loading compared to the traditional polymer-based ASDs. One good way to screen a set of polymers and narrow them down to the most promising candidate is the solvent shift method. This method was first described in the mid-2010s. In this approach, the API is dissolved in a polar solvent, most often a DMSO, and then a small aliquot is introduced into an aqueous medium to generate a supersaturated solution. The API concentration is then monitored by UV over time. Different polymers are tested in parallel, and the effect of each polymer on delaying crystallization is determined at the end. In this example shown here, you can clearly see how commonly used polymers performed compared to a blank sample without polymer, with HPMCAS medium grade and high grade emerging as the best stabilizer. Typically, the solvent shift method is carried out using the Inform or Rainbow system from Pion. However, this approach comes with some limitation. The compound must be UV active, and it requires a relatively large amount of material since the working volume is around 40 milliliters, and the overall throughput can be somewhat limited. Here you can see an image of Inform platform with a vessel positioned in place. Inside the vessel you will notice the UV probe, the pH electrode, and the stirrer that ensure the system is properly controlled. The first step in the experiment is to establish a baseline by evaluating a supersaturation ratio without any polymer present. For our study, we began with a supersaturation of 10. To do this, a stock solution of Piroxicam is prepared, and the instrument precisely distributes the required solution to reach the target ratio of 10. The concentration then is monitored in real time, and once we confirm that the supersaturation is appropriate, the next step is typically to repeat this experiment in the presence of polymer. Unfortunately, in the case of Piroxicam, we were not able to obtain a reliable concentration measurement due to a spectral blackout. We attempted measurement with a 1 mm probe, but we were still unable to capture the crystallization process. As a result, we could not apply the solvent shift method to Piroxicam using our platform. We then asked ourselves, could the Crystal16 be used as an alternative for solvent shift experiments? The Crystal16 is a powerful tool. It's a heating block with 16 positions, each equipped with an optical sensor that can directly detect the presence or the absence of solids in suspension. This opens the door to running multiple experiments in parallel. In our case, we tested 4 supersaturations without polymers at different ratios. We prepared a DMSO stock solution of Piroxicam and added different volumes to reach a supersaturation of 5, 10, 15, and 20. What's really exciting is that compared to our Inform experiment, the Crystal16 uses about 40 times less material, and we can still see precipitation events. Here I've included the graphs to illustrate what's happening. At 0% on the y-axis, we have a solid suspension. If we zoom in on the first 33 minutes, the difference between the supersaturation levels becomes very clear. At a ratio of 5 without any polymers, the solution remains stable for hours before precipitation starts. In contrast, at a supersaturation of 10, we see precipitation beginning just 6 minutes after the addition. And for the highest ratio of 15 and 20, the precipitation occurs much faster within 2 or 3 minutes. Based on the results, we decided that a supersaturation of 10 strikes the right balance. It's fast enough to reveal differences in stabilization, but not so fast that everything crashed out before we can observe the effect of the polymers. Now, let's move on to the screening of a set of 15 polymers. Each polymer was first added to the aqueous medium. We selected a phosphate buffer to maintain the pH close to 7.4. For each experiment, we used about 4 milligrams of polymers, matching the amount of Piroxicam introduced from the DMSO stock, while keeping a supersaturation ratio of 10. A library including a diverse range of polymers, and we also decided to test casein hydrolysate as a protein-based excipient. The results were quite different, with some excipient failing to maintain the solution, with precipitation occurring almost immediately. Other extended stabilities for several hours before crystallization began. At the end of each experiment, we recorded visual observation of the samples. This gave us valuable insight into how much Piroxicam had precipitated, so we could rank the polymers in terms of their abilities to stabilize the supersaturation state. On these graphs, you can clearly see the effect of each polymer on stabilizing the supersaturated solutions, as well as the amount of Piroxicam remaining at the end of the experiment. To highlight our key finding, I've marked with a star the polymers that shown the most promises. They are the candidates we selected for a deeper investigation. Here I have put a zoom-in version with a PVPVA, which looks possibly the best polymer. So, how do we actually prepare amorphous dispersion? There are several techniques available. One option is solvent evaporation, where the API and the polymer are dissolved in an organic solvent and the solvent is removed, for example, by rotary evaporators. A related approach is spray drying, which is widely used because it allows for efficient solvent removal. Lyophilization can also be applied, although it's more limited since only a small number of organic solvents are able to sublime. Another technique is co-precipitation with anti-solvent, which we use in-house, though it doesn't always work for every API. And of course, hot melt extrusion, which is very popular in the industry, provided the compound can melt without decomposing. Ultimately, the choice of method depends strongly on the physical property of the API, especially its solubility in organic solvent and thermal stability. For Piroxicam, we selected spray drying as our method to generate ASD. This is not only a scalable process, but also one of the most common techniques to produce commercial products. So, what is the principle of spray drying? First, the API and the polymer are dissolved in a volatile solvent. The solution is pumped into the nozzle of a spray dryer, where it is atomized into tiny droplets. These droplets are rapidly dried as the solvent evaporates, leaving behind solids and spherical particles. The particles are then separating from the drying gas into a cyclone, collected into a container at the bottom, while the vapors are condensed and the drying gas is recycled into the system. At our Cambridge site, we have a Bookie B290 mini spray dryer. It is fully enclosed, so we can work with potent APIs. We have a high-performing cyclone and a modified collection pot, so we can work with quantity as low as 200 mg if needed. In our case study, Piroxicam, and the polymers are dissolved in a mixture of water acetone. The samples are spray dried and further dried at the end overnight in the vacuum oven. And I have put here some of the parameters we used. We spray dry 3 different polymers: HPMC, HPMCAS medium grade, PVPVA 64, each with a drug loading of 20%, and then we carried out a full characterization of the resulting materials. With HPMC, we unfortunately obtained a poorly crystalline solid, which indicated limited success in stabilizing Piroxicam in the amorphous state. However, with HPMC medium grade and PVPVA, we achieve a fully amorphous phase, each showing a single glass transition temperature, 84 and 96 degrees respectively, confirming good miscibility between drug and polymer. We did detect small traces of residual acetone by NMR, which we plan to quantify more precisely by GC analysis. To test stability, we expose these dispersions to humidity stress for 2 weeks in open vials, and encouragingly, no crystallization was observed. Even more promising, after storing the sample in closed vial in our lab for over 1 year, we did not see any sign of recrystallization. Finally, we evaluated the performance of the amorphous dispersions directly against the crystalline Piroxicam. For this, we use our Inform platform, which allows us to adjust the pH dynamically over time to simulate passage through the gastrointestinal tract. This is particularly important for Piroxicam since its solubility changes significantly depending on the pH. These are the results of a free powder dissolution. The experiments were run in duplicates. Here we can see that at a low pH, the amorphous dispersion rapidly dissolves with a preference for PVPVA. To summarize, we presented Piroxicam as a model compound. It is a poorly soluble BCS Class II compound, and we gave a reason why it could benefit from ASD formulation. We show you the principle of the solvent shift method as a screening tool to reduce the number of polymers. We talk about the different Crystal16 experiments we could use, the supersaturation selection, and the polymer screen. We then selected 3 polymers and describe the ASD preparation using our spray dryer. 2 out of 3 spray drying experiments gave successful ASD. These were then fully characterized, and we showed you how the ASD were better performing over the low pH range, especially the PVPVA. So I hope you enjoy this talk on amorphous dispersion. I would like to thank for your attention, and I would like to thank the ASD team in Cambridge as well as the analytical team, especially Christina Kashia, who did the Crystal16 experiment, and also Amy, who did the free powder dissolution. I'm happy to answer any of your questions now.
Thank you very much, Thierry. A beautiful case study that you have presented. And it's nice to present also on amorphous solid dispersions, because we always, of course, in development of a drug or when we have to work on a drug, we always go the let's say easiest way: polymorph, salt, co-crystals, and amorphous solid dispersions are our last option. But beautiful to see also from this perspective. And also the Crystal16, I was very happy to see that even with more yellowish type of solutions, you had good results, Thierry. These are sometimes questions that we receive from also our customers, how the Crystal16 behaves with colors, and so a nice example that still depend also with other type of solutions, not only transparent or white, that the Crystal16 was and still is a powerful tool now for many years on the market. Thierry, we have a few questions coming here, but I wanted to ask you, how many projects do you have? Often many projects on amorphous solid dispersions nowadays. How is a bit the development in the in the pharma industry? Do you receive in these cases?
That's very popular when people they are getting to a point where the molecule does not dissolve. And if you look at some of the recent figures, I think a lot of anti-cancer drugs, they got these solubility problems. So I would say there is more and more anti-cancer drugs going on the market, which are actually formulated in as amorphous dispersion. And as we get to see quite a lot of drugs passing our way, we have some of these drugs coming our way, and sometimes that's what we would suggest to solve the solubility problem. So yes, that's something we try to solve for clients, especially sometimes you want the exposure to the animal as part of the animal studies, just to see any negative effect, especially when people they go to look at 10 times the dose for testing the toxicity of the material. So they need that exposure, and sometimes the amorphous dispersion is the only way where you can get a decent exposure at higher dose.
And as methods, what you use, you have showed a few methods, but what works best? Of course, is also compound dependent, but what are the few methods for amorphous or dispersions that you say, okay, as a scientist or my team goes first, they will always try X, Y, Z. What would be your advice for our attendees today?
I think as part of R&D, you need really to understand your compound, how it's behaving, what's the property. For example, for Piroxicam, we could dissolve it in more than one organic solvent. Obviously, DMSO was used, but there is other options. And that's not the case for every compound. So I remember working a few years ago on a compound where even DMSO was not something we could use. So that's really limiting what you could do to make amorphous dispersion. And in this case, if the material has like a clear melt, maybe you can go to that high temperature and actually introduce the polymer at this stage, because obviously for anything solvent-based related, you need to dissolve your samples. Obviously, if you can't dissolve in anything, you can't do co-precipitation. You need to dissolve your sample to make spray drying experiments. If you can't do that, you need to look at hot melt. And maybe after that, you may want to look at co-grinding experiment where you will co-grind your sample with some polymer just to see if that sort of dry method can make your amorphous dispersion. So just in a nutshell, it's really depending on the property, and you can't really decide unless you have a good understanding on the solubility, melting point of your API.
And besides the different, of course, solvents that you have used and different polymers, I guess you guys use also different drug loads and different percentages of polymers, right? How do you usually design if you look more
So I think sometimes especially because there is many polymers on the market, it's good to do like a similar drug loading to compare with. And typically, if you look at what's on the market, obviously precedent, a good starting point is possibly going at 20% drug loading. And if you've got obviously some good candidate, you can go take this good candidate, explore them further with different drug loading to see if you can increase it. So I would say a starting point is good for 20%. And after that, if nothing comes out, you may want to explore again another screening set with either more polymers or maybe set at a different ratio. But typically with a 20%, you may eliminate some polymers. And after that, you have a reasonable number to increase the drug ratio if that's an option.
Okay. I actually covered this. We had a question related to the different composition ratios of the polymer tested with the API. So I guess it was a question related to different ratios of the polymer. We have another question from your slide 10, and there you were saying something about about the equilibration time. So and the question is related to, are 24 hours enough to reach solid liquid equilibrium for such compound having a low solubility in the solvents used? Well,
yeah, I think possibly that would be to explore a bit further. I mean, if you look at Piroxicam solubility in the literature, I don't think people they agree on one solubility. I've seen some figures, possibly up to twofold difference in terms of solubility. So typically 24 hours, that's quite standard in the industry, but maybe there is something to look, are we going to find a difference at 48 hours? And the question is, where do we stop in terms of obviously looking at the time for solubility? Obviously, if you think about relating to the drug in the body, the digestion time is between 23 and 35 hours. So again, 24 hours is within that sort of time frame, knowing that in the GI tract, for example, the compound will have only 2 hours to spend in the stomach, something like up to 6 hours in the small intestine. So 24 hours is really indicating that the compound is not soluble and has some solubility problem, obviously, when it's related to the body. But is 24 hours enough to reach equilibrium? I think unless we are taking it further, I don't know. It's really compound dependent.
Yeah, indeed. And I do recall from many years back when I was also doing quite a lot of projects for pharma companies, that also companies have also their own, not to call them regulations, but workflows that not necessarily driven by specifically by a compound or so, but also standard times of 24, 48 hours, 72 hours, and so on, that they would, but indeed, if the drug stays only 2 hours in the
stomach, yeah.
Yes, there's 24 hours, plenty of time, more than enough, right? Yeah. So that's also something to take into account when you do this type of experiment. We have one more question. How did you go about selecting the right polymers to screen against? And how can you check their acceptability? They are acceptable.
Yes, so we will go with actually polymers which are in the FDA website, there is something called the IIG list, which is any excipient which has a precedent is listed in that list. So we will select as a priority things where there is a precedent. So that's how we would select initially polymers or things we screen against. And obviously, there is always more. So, in the case of a Crystal16, we would put one blank and we'll use 15 accepted polymers or excipients. If nothing's coming out, there is other things we can screen, but a sort of second list will possibly have either a less precedent on the market, but still be pharmaceutically acceptable, or if it's something which has been designed to be a polymer for amorphous dispersion where there is not a precedent on the market, we would like to know, is it something that people are using to develop a drug right now, and is it going to be on the market soon? So obviously everything is how much risk do you take to take something novel? I would say big pharma company, they may take that forward. Small biotechs, they may want to actually use something which has some precedent, just because obviously the toxicology study hasn't been done on some of new material which people are developing for amorphous dispersion.
Yes, and I have also a question to you, Thierry, about large scale. So you have nicely showed to us quite a few things about how we can get to those amorphous solid dispersions and so. But what is your take on scale up of amorphous solid dispersions? And I see also a question, a bit more detailed, but if you give me your opinion about, how would you, how challenging do you think it is, and what is your take on it?
Yeah, so spray drying is a good technology to use because it's available pretty much quite widely now. The instrument we have is a small scale bookie that is there everywhere. There's a lot of literature about it if you look at dissolving your polymer. So that's a quick way to get the material. And when you look at how amorphous dispersion were made 20 years ago, that would be possibly the way forward. But things have evolved. And in terms of cost, and in terms of commercially viable amorphous dispersion, a lot of people are moving to hot melt extrusion, which is another good way to scale up the amorphous dispersion. I would say that's possibly less available with formulation company. So I think it's really depending where you are in your pipeline. If you want to do screening, scale up something quickly, you need to look at what's available. But when you go commercially, you need to consider obviously spray drying, hot melt extrusion, but you should not forget things like co-precipitation. So co-precipitation is a good technique to explore because a lot of CDMO, they've got large vessels where they do their chemistry, and that could be done in the same equipment. So you end up putting a solution into an anti-solvent to precipitate, you filter it. But that would be by far the most economical way of getting the amorphous dispersion, but it doesn't work all the time, especially as you're increasing scale. Kinetic could be challenging, but that's something as well to consider. And there is products on the market as well using co-precipitation.
And what would be your few parameters that, and is related to the question that we have received now, what are the parameters that are critical to focus on when you go from milligram to large scale? What are those things that you look at first?
So I think if it's to make amorphous dispersion on a small scale, you could obviously use the spray dryer, and we can get really small amount on the spray dryer. Because spray drying is a continuous process, I would say you generate material all the time through the nozzle, and that's quite scalable. However, if you look at co-precipitation, some of the parameters are more challenging because on a larger scale, the kinetic of precipitation would be different, the kinetic of mixing would be different. So these will be the parameters to look when you look at co-precipitation. When you work on a hot melt system, again, that's continuous. You're feeding your hot melt exclusion, you've got material coming out at the other end. So typically, a small scale equipment, if you've got a process, they will scale up fairly well. So I don't think there's many challenges which can't be solved, but possibly the co-precipitation is where you need possibly to look at modeling, shear forces, kinetic of precipitation. So a bit more modeling is involved when you look at co-precipitation.
Yeah. Thank you very much, Thierry. I think we are at the end of this webinar. Thank you for sharing this case study, your experience and your knowledge with our attendees today. For those that they are at home, please, if you have questions, send them to us still at info@crystallizationsystems.com, or maybe Thierry, you would like to share your also email address?
So it's at the beginning of the presentation. So okay. It's Thierry.Bonnaud@veranova.com.
Okay, and we will share also the slides and the recording with everyone, so you could also contact directly Thierry if needed for more information. Thank you once again, Thierry. It was a pleasure to have you again with us today, and thank you all attending today. Stay tuned. Beginning of November, we have another webinar coming soon on crystallization systems. Thank you all. Bye.